So, you know, I think it's funny that you talked about tumor heterogeneity because granulomas are a lot like tumors. And so I try to go to as many cancer biology talks and you know, the oncology presentations as I can, because there's so many things that tuberculosis I think that we learn from tumor biology. So it's a very, very appropriate analogy. So thank you for the invitation to talk. I appreciate the opportunity to present the work that we're doing on tuberculosis and how tuberculous granulomas are organized by cytokine expression. This is all maybe a very surface level presentation on some level, but I hope that it makes you guys think there's a lot more that we can do with this, because I think that the field of tuberculosis needs more emphasis on some of the computational systems to really understand what's happening in this environment. So I'm just going to start off talking a little bit about tuberculosis. Not everybody's familiar with it, but TB is something that's a problem worldwide. And TB is caused by Mycobacterium tuberculosis infection. It's an acid-fast bacilli. And it's estimated that about a third of the world's population is infected with this bacterium. So that equates to about 2 billion people that have latent infections. Most people are very good at being able to protect themselves against active disease. And of these 2 billion people that are infected, there are about 9 million new cases every year. These are 2013 statistics, but that would change a lot in the last couple of years. And of those 9 million new cases, there are about 1.5 million deaths on an annual basis. And TB is one of the top killers of women and actually young men also within the ages of 15 and 44 years old. So it's an incredible problem with people that are at the peak of their productivity in their communities. And so you can see some areas are particularly hard hit by tuberculosis. You can see that sub-Saharan Africa is an area with a significant amount of endemic TB. And then Southeast Asia and East Asia and the old Soviet Union are particularly problematic for TB. And multi-drug resistance is becoming a problem. And extensively drug resistance is becoming a problem. And there's even completely drug resistant tuberculosis. One of the major drivers of all this is the HIV epidemic. And HIV-infected people are more likely to die from tuberculosis than anything else. And even people with normal levels of CD4 T cells, which is oftentimes looked at as the prognosticative factor in HIV-related disease, those individuals are also at high risk for active and reactivated TB. So there's something about TB and the host immune system that really is highly interlinked. But you know, I want to reinforce that most of the people that are infected by this bacterium never know they're infected. So these people have latent infections, and you never know, they'll never know that they have the bacterium unless they are given a skin test, perhaps. And although they're infected and never know it, these people can become are susceptible to reactivated tuberculosis under certain circumstances. So if you have immunosuppression, let's say you're on Humira, if you watch those Humira commercials on TV, you see several mentions on there, don't take this if you have TB, get skin tested for TB, et cetera, before that. So immunosuppression is a real significant source of reactivation. Age, poor health. So if you think about people that are significant stress in individual and communities that are underserved, these people are oftentimes at risk of reactivated TB. And then again, HIV is the most strong factor in predisposing an individual to tuberculosis. So TB is treatable. There are drugs that can treat most cases. Unfortunately, the drugs are terrible, and they have severe side effects, and you have to take them for a long time. So you have to take 2 to 4 drugs for at least 4 to 6 months, and that makes compliance a problem. And so people that are less likely to be compliant are also more likely to develop drug-resistant strains that gets transmitted throughout the community. And so one of the real problems in tuberculosis is this issue of drug resistance. So how do you study tuberculosis? Well, it's a difficult disease to study. So a lot of the animal models that we use for other diseases, mouse models and so on, are not very representative of human pathology. They have different granuloma forms, different types of inflammation, and the course of disease is completely different. One animal that does have very, very similar disease to what we see in humans is a cynomolgus macaque. And this is a small non-human primate. And and it's the best characterized model of latent tuberculosis and one of the best models for tuberculosis in general. And this animal has the full spectrum of disease, not just active disease, not just chronic disease, but completely latent disease. And the symptoms are the same as what we'd see in humans, for the most part, clinical symptoms at least. Weight loss, coughing, pulmonary involvement. And so these animals can be latent for years. We can treat them with Humira or other immunosuppressive drugs, and they can reactivate. You can also infect them with SIV, an HIV-like virus, and they'll experience reactivated disease. So they're very human in that sense. And most importantly, the pathology of these animals is very similar to humans. And TB isn't generally a disseminated disease like a lot of other bacterial infections. So tuberculosis localizes itself within a single site of infection called the granuloma, so in the lungs usually. And the other thing about this animal is we can follow these granulomas from their inception, basically at the very early stages of disease, to the full life history of these granulomas. So I come out of Joanna Flynn's lab, and through her studies, we have thousands of granulomas. And we have granulomas from all sorts of different time points and pathologies and tissue sites. And a lot of these granulomas, we have bacterial burdens that we know. We've used PET-CT to measure their inflammatory profiles. And we've got all these granulomas from different treatments. And all these granulomas say something different. And maybe one of the best ways to find out what works in TB, what's protective in TB, is to figure out what's not protective first. And so we're still, I think, in the process of figuring out what's the basic biology of tuberculosis at the site of infection. And these particular treatments will be useful in figuring out where does it go wrong. So now I just want to talk a little bit about granulomas, because this is sort of the heart of the matter in tuberculosis. You develop granulomas after infection with tuberculosis. That's one of the hallmarks of this disease. And you develop, or you become infected when your neighbor who's got bacteria in his or her airway coughs those bacteria out. You inhale them. Those bacteria are phagocytosed by an alveolar macrophage, where they grow intracellularly. And for the first couple weeks of infection, it's been demonstrated that only the innate immune system is responding to this infection. So you've got aggregates of cells, mostly macrophages and monocytes and neutrophils, forming in this localized area. But at some point, a dendritic cell primes T cells in the lymph nodes. Those T cells migrate back to the site of infection. And the adaptive immune response is really what starts to control the bacterial infection. And that initiates formation of this granuloma. So granulomas are multicellular structures. They're fairly large. And this is the place where both the anti-mycobacterial response occurs, but it's also the place where the bacteria lives. So this structure plays two roles. So this is a prototypic caseous granuloma, a non-necrotic granuloma, you'll sometimes hear it referred to as. So here's a region of caseum. This is a poorly defined cheese-like material. Around that, these are epithelioid macrophages. And then just outside of that, this is the lymphocyte cuff. And then you can see this adjacent uninvolved lung that's largely separate from the site of infection. And each one of these regions has a different complement of cells. So this lymphocyte cuff region is where the lymphocytes are most abundant. So lymphocytes, T cells, B cells, NK cells. You find eosinophils out there. But there are also macrophages out there, and there are neutrophils in there. And this is where fibrosis oftentimes occurs, this is where blood vessels are, and then there's some residual epithelial surfaces that you can see in this area. So the macrophage region is dominated by a particular type of macrophage called the epithelioid macrophage. And these are a particularly derived form of macrophage. We don't really understand their origin, though, but they're quite a unique population of cells. And these cells are oftentimes infected with Mycobacterium tuberculosis. So the bacteria can be found in here. And a lot of the bacteria can also be found in the caseum. So this is that dead necrotic area. And then between the epithelioid macrophages and the caseum is this really rich, a region of really rich neutrophil abundance. And so all of these different regions have some interactions, and we're trying to figure that out.
How long does it take for that architecture to configure after start?
Yeah, that's a good question. So the earliest that we find granulomas are about 3 or 4 weeks, and by 3 or 4 weeks, that architecture is already formed. So it's a rapid event once it starts to occur. But before that, we can't really detect that, either by the standard histologic techniques. We can't see it by PET CT. So really everything that happens before that is kind of a black box. So this is a cross-section of a granuloma, looking at things in a little more detail. So here you can see the caseum. Again, kind of an amorphous region. You can see neutrophils scattered throughout it. A lot of them are necrotic. Outside of that, this is the epithelioid macrophage region. So these cells have a large or a high cytoplasm in the nucleus ratio. They're big cells. And you can also find in this region, and in the lymphocyte cuff, these Langhans giant cells. Langhans giant cells are oftentimes infected with Mycobacterium. And although they're not indicative of tuberculosis, they're commonly found in granulomas. So here's the lymphocyte cuff. Again, lots of smaller lymphocyte-like cells. You can see some eosinophils, obviously staining out here. But then you can see there are macrophages in this environment, and there are neutrophils that are a little harder to find. So granulomas aren't static. They don't just form and exist. Granulomas change over time. And one of the tools that we've got to look at them is a PET CT in the VSL3. And so we can inject these animals with a glucose analog, FDG, and this radiolabeled tracer is preferentially taken up by glucose-utilizing cells. And there are a lot of those cells in granulomas. And so in this section of monkey lung, what you can see is a pair of granulomas. So one granuloma starts out hot, and if you watch over time, it shrinks in both its uptake of glucose, but also in size. Whereas this little granuloma down here begins off fairly diffuse and fairly dim in terms of its glucose uptake, and then increases over time. And these granulomas are separated by centimeters. And so these each have different and independent trajectories. And you can see with this graph here, just looking at 5 different granulomas, they kind of go all over the place. So one granuloma starts off here and ends up here. 2 actually start off here and end up here. And then there's one that starts off here and ends up lower. So what accounts for that and what that results in is a great interest in the world of tuberculosis. So while I'll talk a lot about caseous granulomas today, there are a lot of different variations in granulomas. And a lot of what you're seeing today, again, is going to be a snapshot taken at the end of the study period for this animal. And so these granulomas are all constantly changing. And in the end, when we look at it, we find a number of different phenotypes for granulomas. So you have the non-necrotic granuloma, the caseous granuloma we've been talking about, or a suppurative granuloma that's completely filled with neutrophils. And then there are non-necrotic granulomas. This granuloma is nothing but epithelioid macrophages. There's no caseum in there yet. And then this is what you would typically associate with a good response. So an animal that has been able to take care of the bacteria, been able to kill those bacteria, will have lesions that start to resolve and become fibrotic and mineralized. And over time, these lesions can go away and just leave scars in the lung.
Josh, quick question. What is the origin and the chemical nature of this cheese-like material that's found in the product?
Well,
I don't know, is this being characterized in any way?
Yeah, people are working on it, but it's a really complicated environment. And part of the problem is, first off, you can't get casein easily from humans because you can't easily access human granulomas.
Okay.
And then in monkeys, you can actually do some of that work. So there is some proteomic work being done on that. But if you look at if you do immunohistochemistry in a granuloma, you can see things that are staying in there. There are a lot of remnants of neutrophil. Neutrophils and neutrophil-related products in there. It's full of like MMPs, there's a lot of DNA in there, a lot of lipid material that's probably associated with necrotic cells.
So it could be that, the supportive could become the necrotic evolutionary-wise. I don't know.
It's possible. Yeah, we don't really know because it's we have been trying to draw some tentative directions for a granuloma's life history.
Yeah.
But every time we feel like we do that, the pathology argues against what we've tried to say. So we're trying to figure all that out still. Yeah. So.
Do you see the supportive ones in the macaque model?
Oh yeah, yeah. All these are from macaques. And usually if you find a suppurative granuloma, that's a bad thing. So there's a lot of evidence suggesting that lots of neutrophils are detrimental for protection in TB. And there are a lot of reasons for that, and that's one of the main focuses of my work. But neutrophils do a lot of things. They produce a lot of proteases that interrupt cell-cell interactions. They produce cytokines. They do all sorts of things that are poorly regulated, and I think part of that regulation is one of the reasons why neutrophils are so bad. So where are the bacteria in all the system? Well, they're in a number of places. So here you can see the H&E, and this is the auramine rhotamine staining showing where the bacteria are in these granulomas. So in this granuloma with a lot of neutrophils, these bacteria are living happily next to neutrophils. In this case, there's a giant cell that's filled with bacteria, another big caseous granuloma. In this case, this is a fibrocalcific granuloma, so this is the granuloma you expect to see resolution occurring in. But this granuloma, even though it seems to be resolving, still has mycobacterial antigen, and they even look like there are intact bacteria. So if you take and find a fibrocalcific granuloma in a cat, you can take that granuloma, homogenize it, and plate it, and it can still grow bacteria. So even though it looks like it's resolving, you still don't necessarily have bacterial killing occurring. And that leads me to this slide. So in this slide, we're looking at different granulomas of bacterial burden per granuloma. So you can see at 4 weeks, there's a fairly high bacterial burden in all of these granulomas. And then after the innate immune system has started to be activated by the adaptive immune response, looking later in infection, you can see that there's a drop in the number of bacteria. So the earliest time points are the points that generally have the most bacteria in a granuloma. But what's really interesting is that if you look at this graph, you can see that so each column represents a different animal. And so every dot represents a granuloma. So you've got granulomas in this one animal here, for example, that have pretty high bacterial burdens. But in the same animal, you've got granulomas that are completely sterile. And understanding why that happens, so why you have granulomas that are protective and have sterilizing immunity versus granulomas that don't seem like they have any protective benefit at all, is a really important thing that needs to be done. And this would benefit vaccine development because we don't really have a good feel for what the correlates of disease or correlates of protection are at the site of infection, and it will also help with understanding how treatment occurs. So one thing we're looking at is this is a hypothesis that balance is important in a granuloma. Balancing a pro-inflammatory and an anti-inflammatory milieu. And we think that the best response is one that balances both too much, both the amount of inflammation and yet maximizes the amount of bacterial killing. So if you have too much inflammation, oftentimes that inflammation just means you have more host cells for bacteria to thrive in. If you don't have enough inflammation, you don't get enough activation and enough killing. And so we're trying to figure out what's happening here. Something else that's becoming more and more apparent is that these responses need to be spatially and temporally coordinated. So an early granuloma probably has different biology and different requirements than a later granuloma. And the cells in the lymphocyte cuff need to do different things in the right time and the right place relative to cells in the epithelial macrophage region. So we're trying to figure out how all this works. And so a lot of what we do is looking at regulation at the site of infection. And that's really where it's at in TB. So this is a granuloma, just showing the position of cells in that tissue. So what we've got is are 3 different populations of cells. So we got CD68 positive macrophages, CD3 positive T cells, and then calprotectin positive neutrophils. And we just identified the location of every one of these cells and plotted it over here to show how nicely organized these are in terms of where those cells are. And so here we've got this abundant population of neutrophils. This is caseum in the middle, epithelioid macrophages out here, and T cells out here. So there's some happy medium for this granuloma in terms of how many of each cell population are present there. And we don't know what that is in this case. Most of the data that I'm talking about today come from animals with active TB. So maybe a lot of what I'm showing is not the happy medium, but it's sort of on the end of the pathologic end. And so we're trying to figure out the other side of the equation. What's the good thing?
These structures have no gaping holes necessarily.
No, yeah, they're if you took a basketball, that's what they would look like. With lymphocytes on the outside and going inside. Yep. So the macrophages are distinct within different regions too. So if you take the granuloma standard for CD11C, CD68, CD163, what you find is there are CD163 positive macrophages out here on the outer region of a granuloma. These are interstitial macrophages. These cells here, these are actually alveolar macrophage-like cells. Alveolar macrophages are CD11C positive, CD68 positive, and CD163 positive. But this region here, this epithelial macrophage population, is strongly positive for CD11C and CD163, and it's dim for CD160. It's positive for CD11C, CD68, and dim for CD163. These regions are also functionally distinct. So if you stain for arginase and inos, these are two enzymes that compete with each other for arginine. And inos is an important anti-mycobacterial factor in tuberculosis. What you find is that these epithelial macrophages are enriched for inos, and the bulk of the arginase expression occurs up here. But there is some arginase expression in those epithelial macrophages. So it's hard to find two poles of the macrophage phenotype. So in primates, macrophages are oftentimes described as M1 and M2. But that's an oversimplification. There's a whole spectrum of phenotypes within that. And so trying to understand sort of the ratio of one phenotype versus another phenotype in its role is a complicated endeavor. So these regions are quantitatively different in terms of macrophage populations. So your epithelial macrophage population is different than your lymphocyte cuff macrophage population. And the CD163 positive macrophages are much more M2-like, so pro-healing or pro-fibrotic macrophage, whereas these guys are your anti-microbial, antibacterial effectors. And then the same phenomenon occurs with the inos and arginase expression. And since these enzymes compete against each other, it's really the ratio of one enzyme to another that differentiates the outcome of the cell polarization. So epithelial macrophages are much more inos-rich than the lymphocyte macrophages. And that presumably has an effect on what happens in that structure. So why does this structure form? What's the driver of all of this behavior? It's reproducible. It seems like it happens all the time. Granulomas are a uniform feature virtually across the animal kingdom. So if you take a frog and you inject it with Mycobacterium marinum, which is a mycobacterium that affects fish, that frog will develop granulomas. And they're all very similar to human granulomas in different aspects at least. So this is a conserved response across many species. So one thing that's obvious would be exposure to mycobacterial antigens. So these mycobacteria secrete all sorts of things that elicit toll-like receptor-mediated responses. Another thing is the cytokine signaling that the cells engage in. So one cell talks to another cell, tells that cell to do something else. That cell interacts with someone else. And so all these things come together. And in the end, we get these structures that are both organized in terms of phenotypic differences by region, but also functional difference by region. So one of the things that we're looking at as a primary regulator of these responses is type 1 interferon. And type 1 interferon might be one of these factors that regulates the pro-inflammatory and the anti-inflammatory side. And trying to find out how this does this and how this interaction occurs is really the focus of much of my work right now. So looking at type 1 interferons in tuberculosis, there are a whole host of different type 1 interferons. They all signal through the interferon alpha receptor 1 receptor. And when you think about type 1 interferons, you think about plasmacytoid dendritic cells as primary expressors of it, especially in HIV. But epithelial surfaces, because they're sort of the first line of defense at the mucosal surface in viral infections, are also strong expressors of type 1 interferons. And type 1 interferons are really important for antiviral responses. So they're critical on that. And this includes HIV and SIV. But again, that concept of balance is really important. So if you don't have enough, it's a problem. If you have too much, there are other pathologic consequences. So type 1 interferons in all these other systems have been identified, and their balance is being considered. In TB, type 1 interferons are associated with bad outcomes in TB. And that's as a consequence of a number of different things. So they're linked with downregulation of IL-1 beta, and IL-1 beta is an important anti-microbial factor. They are responsive or macrophages are responsive to them, and they can drive inflammatory responses there. And in humans with active TB, there's a peripheral blood signature that's associated with neutrophils, and has indicated that this is driven by type 1 interferon. So all of this really is done either in periphery, so in the blood or the BAL, but it's hard to know what's happening at the site of infection. And again, this is where we have a difficult time in tuberculosis, just because we don't have an abundance of human granulomas to look at. So the question I have is, what cells express type 1 interferon? Who is type 1 interferon influencing in this situation? And first off, why is type 1 interferon being expressed at all? And this is all occurring at the site of infection. So it's clear that from ELISA data that if you compare normal blood to a granuloma and normalize it to the total protein in those samples, granulomas are enriched for type 1 interferon. So there's, and this is measured by interferon alpha 2 ELISA. So granulomas appear to preferentially express type 1 interferon or have upregulated type 1 interferon. So we're going to figure out where that occurs in the cells, and primarily use immunohistochemistry today in our discussion today to do that. So some relevant background on type 1 interferon and type 2 interferon signaling, because interferon gamma is a critical factor in the microbial responses and protection against TB. These two cytokines both go through STAT1, but we can differentiate type 1 and type 2 interferons by looking at STAT2. So only type 1 interferons go through STAT2. And we could also look at interferon IFNAR1 or interferon gamma receptor expression on cells. And complicating the issue to some degree is the fact that interferon alpha beta can also go through the phospho-STAT3 pathway. So if we look at this in a caseous granuloma, this is what we see, and this is very typical architecture. So this red staining, this is the phospho-STAT2. This blue staining, this is phospho-STAT1. And green in this case is CD11C. So these are your epithelioid macrophages into here. This is caseum. Those macrophages appear to export the CD11C into the caseum. And so you can see that this outer region of the granuloma is enriched for the phospho-STAT2. And if you look at the phospho-STAT3 component in a different granuloma, one that's rich in neutrophils, you can see a similar pattern developing here. So the outer regions of this granuloma are strong in phospho-STAT3, whereas the epithelioid macrophage populations are strong in phospho-STAT1. But these neutrophils are negative for both phospho-STAT1 and phospho-STAT2. So clearly, neutrophils aren't responding to either interferon gamma or type 1 interferons at this level. But that just looks at signaling. So a number of things could go through phospho-STAT3, for example, IL-6, IL-10, interferon alpha, and a whole host of other things, I suspect. But if we look at just the protein, so this is interferon alpha 2 in a granuloma, what you can see is this population of phospho-STAT3 positive cells is outside of this population, epithelioid macrophages, that stain strongly for interferon alpha. And the population of neutrophils in here is very, very different from interferon alpha in this case. So it appears that epithelioid macrophages are making interferon alpha, and the cells outside of that population are responding to it. Now, the question is, why are epithelioid macrophages doing this in the first place? One possible explanation is through toll-like receptors. So epithelioid macrophages express CD14. We don't know the full complement of toll receptors that they express, but presumably they express things that macrophages typically express. And mycobacteria secrete a lot of antigens. So they're secreting antigens in the caseum, they're secreting antigens while they're intracellularly existing. And these antigens presumably, again, interact with, or have the potential to interact with toll-like receptors. And this results in phosphorylation of IRF3. Phosphorylated IRF3 translocates to the nucleus, and this capacitates type 1 interferon expression in these cells. So if we take a look at this granuloma, what we can see is that you've got strong type 1 interferon expression here. You've got it kind of spreading out through here. And then the neutrophil population is in here. And if you look at a serial section from the same granuloma and stain for the IRF3, phosphorylated IRF3, what you can see is that this epithelioid macrophage population that's positive here for interferon alpha is also positive for phospho IRF3. You'll also note that there are cells out here. These blue cells you can see are alveolar macrophages. And so alveolar macrophages are also positive for phospho IRF3. But when you think about an alveolar macrophage, these cells are constantly moving through the lung. They're exposed to all sorts of different antimicrobial products, or microbial products, I should say. And so it should be no surprise that alveolar macrophages express all sorts of different factors that may be related to signaling through toll-like receptors. So type 1 interferon expression should be expected in those cells. So we take a closer look at that granuloma and just look at this region of neutrophils. And here we can see the nuclei from those cells. You can see that these neutrophils have, to some level in the population, phosphorylated IRF3 in their nuclei also. And from previous data, we've shown that neutrophils can produce type 1 interferons and do produce it in the granuloma, although it's probably considerably less than what you're seeing in all these epithelioid macrophages. But it's an important observation that some of the neutrophils, not all of them, are phospho IRF3 positive. So now if we zoom out and we take this granuloma here and we look at this in the greater tissue context, what we can see is that there's a lot of phosphorylated IRF3 in this area. This area here, this bright pink area, that's ciliated epithelium, and those cells should be producing type 1 interferon. You can see several other granulomas in this environment. So there's one here that's eroding into an airway. You see ciliated epithelia here. There's another granuloma here, and there's a third one that's just adjacent to this airway. And so if you look at the heterogeneity within this piece of tissue, so these granulomas are separated by millimeters. This granuloma is clearly a foci of phospho IRF3, probably a focus for interferon alpha expression, and that these other granulomas are variable in terms of what they're doing. So there's a hypothesis in TB called the lottery hypothesis. If you have 100 granulomas, what's the chance of, and all those granulomas are well contained, what's the chance that one of those granulomas goes bad? And that's the granuloma that makes you sick. So if you're thinking about the lottery hypothesis, this might be a granuloma that's poised to go bad. Even though these guys are, this granuloma eroding into an airway, this is the one that infects your neighbor. This is the one that disseminates throughout the rest of your body. So we're trying to understand how all this works in the context of an incredibly heterogeneous system where one granuloma is certainly not necessarily representative of all of them, and somewhere there's a mean state of interferon alpha expression. So that describes where interferons are expressed, where type 1 interferons are expressed. But it doesn't necessarily say anything about where they're signaling or what's, well, the type 1, the stat data suggests where they're signaling. But does that signaling lead to products that we would expect to be associated with type 1 interferons? So there are a couple different ways we can address this question. First off, we can look at the FNR1 receptor and see where that's expressed. Presumably, if you don't express FNR1, you can't be experiencing type 1 interferon activation. The other way of looking at that is looking at interferon responsive genes. So we could look at MX1, which is commonly associated with type 1 interferons, and an important role in viral infections. It's poorly defined, but we don't think it's got a role necessarily in tuberculosis. In contrast, IL-10 is an important anti-inflammatory cytokine that's correlated with exacerbated disease and tuberculosis, but it's also got a protective response too. So we can look at these different factors to try to understand where all this occurs in the granuloma. So again, looking back at this granuloma, you can see the phospho IRF3 staining here. So these are the cells that are making interferon alpha. And now in the red over here, what you can see are the IFNAR1 positive cells. And so if you take overlay these, what you see is that this region of epithelioid macrophages is really dim for IFNAR1, whereas these lymphocyte cuff cells, those are the cells that are IFNAR1 positive. So these are the cells that seemingly could be receiving that type 1 interferon signal. And there's a particular population of macrophages that I'm very interested in that you find in the lymphocyte cuff. So if you look at this granuloma here, you can see, again, epithelioid macrophages, strong for interferon alpha 2. This population of macrophages is really rich in MX1. And the more we look at granulomas, the more we see that these cells appear to have some relationship with the granuloma's bacterial burden and how sick that animal was. So if we take and quantify just the MX1 signal in different granulomas, in an animal that's got TB only, and a similar animal that's got a co-infection with SIV, we can find that those co-infected animals have higher MX1. And some of that is due to this population of cells. And so we're really interested in trying to figure out what the role of these cells in that tissue environment happens to be. So IL-10 is probably more important than MX1. And both T cells and macrophages can make IL-10. And what we've got here is a granuloma stained for, in green for CD11C. These are alveolar macrophages really brightly out here. And then they're dimly CD11C positive in here. And then the red is IL-10. So here what we've taken, we've plotted the positions of all the nuclei in this granuloma. We've plotted the positions of all the CD11C positive cells. And then the red indicates the position of the IL-10 positive, CD11C positive cells within that environment. And so clearly you can see that the IL-10 expression occurs on the outer rim of that granuloma and just inside of the lymphocyte cuff. So why would that specific localization of IL-10 be important? Well, IL-10 has a number of different functions. So one thing that IL-10 is doing is dampening the immune response. So you don't want to have an immune response that runs wild and starts to damage the local tissue. And that's one thing that we think that IL-10 is doing out here. The other thing to worry about, though, is that IL-10 antagonizes interferon gamma. And so if you have less interferon gamma, you have less macrophage activation in the bactericidal direction, all of a sudden the bacteria get a better handle on being able to survive in the host. And so the host really wants to find a balance between too much IL-10 expression that would lead to not enough immune activation and not enough IL-10 expression that leads to exacerbated immune responses, or exaggerated immune responses, I should say, that damage adjacent tissues. So if we look at this granuloma, so this comes from a human granuloma that we have. What you can see is this rich population of epithelioid macrophages is strongly positive for epithelioid macrophages, strongly positive for interferon alpha. There are also a few interferon alpha positive neutrophils present in this granuloma. And again, all of these IL-10 positive cells occur out here in the lymphocyte cuff. So there's a region-specific stratification of cytokine expression, both in terms of the interferon, type 1 interferon expression, and then type 1 interferon stimulated genes. So just to summarize all this and kind of wrap all of this up in about as tidy a bow as possible for kind of a noisy situation and a difficult system to do this with. So all of this starts with an epithelioid macrophage. And this epithelioid macrophage at some point is activated by interferon gamma. This epithelioid macrophage is in contact with caseum, and it's also in contact with mycobacterial antigens. And everything before this is unknown. We don't know what happened before that. But what we think happens is that these mycobacterial antigens induce IRF3 phosphorylation. That results in nuclear translation of IRF3. And that downstream interaction results in interferon alpha production and secretion by that macrophage. So now that macrophage is communicating with its neighbor in the lymphocyte cuff through IFNAR1. That results in STAT2 phosphorylation, some STAT3 phosphorylation possibly, and then nuclear translation, or translocation. And then that results in expression of interferon-responsive genes, such as the MXA, or MX1 as it's sometimes called, or IL-10. And IL-10 is interacting on macrophages and T cells in this environment and can undergo sort of an autocrine loop. But one thing it can also do is it can shut down that interferon gamma-producing cell that's activating the epithelioid macrophage. And this is where things really start to become problematic. And so there's this loop where things need to be balanced and need to occur probably in a loop, but they can't occur in too strong a direction in either direction, in either pro-inflammatory or anti-inflammatory way. So just to conclude, just kind of wrap this up, macrophages have different microenvironments, and these different microenvironments have different phenotypes and different functions. And we really need to understand why this happens, why these organized regions occur, and how these regions contribute to protection in the host. At this point, another interesting question, and probably just as important question, is why do granulomas fail? One way we can do that is look at these perturbed systems where granulomas are more prone to fail. So what influences a granuloma infected with SIV that results in that granuloma not being able to contain the bacterium? If in a situation where TNF is neutralized, and TNF is required for protective anti-mycobacterial responses. If TNF is neutralized, why does that predispose that granuloma to fail? All of these things have value in understanding the whole system. And at present, the TB field is kind of behind the curve in identifying these local responses. We spend a lot of time looking at responses in the blood. We spend a lot of time looking at peripheral markers, and we don't really understand what's happening locally, really where the action is happening. In part because human granulomas are really tough to come by. People don't get lung resections, usually because they have regular TB. If people get lung resections, it's because they have incurable TB that drugs can't treat. And so the pathology samples that we do have come from people that have died from other causes, and granulomas have been found sort of in the process of disease, or the process of autopsy. We don't know anything about that disease before the autopsy, or from individuals who have had lung resections because of severe disease. We also have problems extrapolating data from cell culture models and mouse models. And so trying to understand the whole complexity of the system and translating that back to humans has been difficult. But macaques offer a really valuable tool for that. And this is a system where the biology is very human, where we have human reagents at work. And honestly, I think what we need now is we just need new tools and new ways to look at the problem to help resolve it. So with that, I just want to acknowledge some of the people in my lab. So Dr. Janeko, Han, and Rashmi Abasi, who's been working on some of these projects. The Flynn lab is where I did my postdoctoral training, and where I was a research instructor, and really have provided me with an amazing number of samples. We have an incredible library of TB-related pathology sections over there. And then the Department of Infectious Disease and Microbiology, and molecular genetics, and the Center for Biologic Imaging. So with that, thank you. Any questions?
Question. Is that, I mean, using the monkey system doesn't seem to be like a really high throughput option.
No, it's not. And when we infect an animal, we infect them with a very low dose of bacteria. So we give them anywhere between probably 5 and 20 bacteria normally. Sometimes we end up a little bit higher, sometimes a little bit lower. And these animals typically develop anywhere between, initially they develop basically 1 granuloma for every bacterium you instill into their lungs. But over the course of the disease, those granulomas get disseminated. And so some animals will be necropsied with 5 granulomas. Other animals will be necropsied with 50 granulomas. And so there's certainly no high throughput dimension to this.
So let me ask you, there's all these tissues on chip approaches are developing, liver on chip, skin on chip, and so on.
Mm-hmm.
Is there a lung on chip program that could be potentially?
There are lung on chip. So you can buy both, the pathology sections on lung. We also work with a computational biology group that we take the data we get from TB, they put it in their model called GranSim, and then we try to do simulations based on that. But again, we're hampered by, in many cases, availability of samples. So it's sample availability outside of the macaque world is really difficult, really challenging. And we're very lucky at the University of Pittsburgh to have that.
You mentioned before about tissue culture models. Possibly, how far are we with those, and are they reliable? Are you working on any of them?
So we're starting to kick around the idea of working with some in vitro granulomas. The problem that people have had, there are a number of different granuloma models available. So you take immune cells, you put some in there at different time points, infect them, and all that stuff. And you do get this emergent behavior of granuloma formation in a dish. The problem you have is that the complexity is so minimal. And a lot of the things that are really valuable, like or controversial or interesting, like neutrophils, can't be maintained in that system. So neutrophils have a very short half-life, and you can make a granuloma that might last, as long as you keep those cells fed, weeks. But you can't put neutrophils in there and have them last more than a day or two. So the difficulty in that system is finding the right questions to ask. Finding the right questions to ask. And so it's a system that certainly has some utility, and it's just a matter of finding the right questions.
So in terms of the histopathology and looking at those images, where would computational pathology be helpful?
Well, one thing that we're really good at is figuring out where cells are. So we can say, well, neutrophils are out here. We can stain for T cells, but trying to identify interactions between cells and understanding a lot of very simple metrics. So if you have a cell over here that secretes a cytokine, what's the distance between this cell here and another cell that's showing some kind of response? And how does that, how can that be generalized across different granulomas? And it kind of gets back to the whole question of high throughput approaches. So at this point, sure, we can stain them, we can image them. But if we took and just by basically put a ruler up to this computer screen and say, well, the difference between this macrophage and this macrophage, it would take us 600 years to get good data. But we could take an image, 20 granulomas a day, 2 weeks, have dozens and dozens and dozens of granulomas, and then hopefully we'll be able to take those images, segment them, and then run them through a computational algorithm that says, in this subset of granulomas where we have a low bacterial burden, we've got a mean distance of so many micrometers, whereas this granuloma this granuloma subset that's poorly responsive and has high bacterial burdens, there's this case. And so that's really where things really need to go in TB. We spend a lot of time looking at individual granulomas, but we don't have a good feel for what the big picture is.
Any other questions? Gosh, thank you so much.
Well, thank you.